if anybody would like to grab a last beverage or bit of food before we do fully get started, please do. All right, cool. Thank you all so much for joining us. We're extremely grateful to have you all here to celebrate National DNA Day. For those of you who don't know about National DNA Day, it is celebrated every year on April 25th, so we're celebrating a little late, to commemorate the publication of the DNA double helix in 1953 and the completion of the Human Genome Project in 2003. And then every year, as a result, we're celebrating advances in genomics and the ways that it's influencing our daily lives. So for us here at Genspace, naturally, DNA has a big role in the work that we do. Who here has never been to Genspace before? This is their first visit. Oh, lots of hands. Wonderful. So for those of you who are unfamiliar with us or what we do, we are a community-based biology lab. That means we're all about open access science, and we want to make space for people, especially from kind of non-scientific backgrounds, to come here to learn science, to create something new, to grow as a human and as a creator, as an entrepreneur, as a curious citizen, to have a space where you can call science yours. So that's really fundamentally what we're about. So we do things like classes and workshops to help teach people some basic skills in science. Frank here has taken some of our classes. We also have a membership program. So Sharon, I know, is one of our members here at the lab. So she comes here and helps work on some scientific projects that are going on. And then we have a smattering of education, outreach, public events, things like this. Or in a couple weeks, we're going to be at the Brooklyn Bridge Kite Festival collecting microbes from the air. So please check out our website and our social media to stay in touch with some of those things that we do. So tonight, we're extremely honored to welcome our fantastic guests, Carl Zimmer and Dr. Janina Jeff. I'll quickly introduce them, although probably you know all about them. So Carl Zimmer writes for the Matter column for the New York Times. He's the author of 13 books. Who here read Game of Genomes on Stat? Anybody? Yeah, sorry, we got a couple of other awesome nerds here. To me, this was some of the most exciting writing about personal genetics and really diving into what does our genome tell us and what can we learn that's ever been done. So it was really cool and exciting to see that in public spaces. Carl's latest book, She Has Her Mother's Laugh, The Power, Perversions, and Potential of Heredity, has won a bajillion different types of awards and nominated for all of the best book lists. The New York Times Book Review named it their Notable Book of the Year. Publishers Weekly picked it for their top 10. The Bailey Gifford Prize winner and The Guardian named it their best book in 2018. So we're super excited to have some copies here that Carl is going to sign after the event. So please pick one up and give it a read if you haven't. It's fantastic. And then our interviewer slash bioinformatician in residence here, Dr. Janina Jeff. She's a human geneticist and the first African American to graduate with a PhD from Vanderbilt University. So snaps for that. She is a senior scientist at Illumina, which is a biotech company that makes all the sequencing machines that we use to analyze all of our genomes. Her research career has been around genetic loci that affect complex common diseases, especially in admixed and African American populations. And she's been involved in like a bunch of amazing scientific studies. Dr. Jeff has also taught lots and lots of classes for undergraduates and graduate students in genetics and statistics. She's taught K-12 science courses, science classroom programs, and she's a STEAM activist, so also with this art and design kind of interest that we have here at the lab. And she's speaking nationally all over the place, amazing, amazing things. She's also a podcaster, so her podcast will launch soon, maybe in the fall, is that right? Yes. Soonish. Her podcast is called In Those Genes, about lost identities in African Americans and genetics. So please join me in welcoming Carl and Janina. Thank you, guys. Oh, okay, I'll go try to let them in. Thank you.
Hey.
Can you guys hear us? Oh,
No.
No? I didn't know if this was a microphone for us to project or a microphone for us to talk about.
Just our booming voices.
Yeah. So today we're talking about the power of knowing your own genome and the journey of going through your genome. And so, Carl, in your book, you talk about your experience of sequencing your genome and what you learned. So I think we should start there. Do you want to give a summary about kind of how you started that journey, what made you want to do that journey?
Yeah, I mean, it really kind of started with just a phone email, I guess. Come in. Should be some chairs. I got an email from a geneticist who I'd written about in the past, and he said, hey, you want to get your genome sequenced? And like, I'm just looking at the email, and I've been writing about DNA sequencing at this point for over 20 years, and I'm like, what? Like, you know, because like, you know, I remember when it was a big deal when the first genome of bacteria was sequenced. Everyone was like, oh my gosh, you know, and then the first genome of a fruit fly. Whoa, and then the first genome of a human, and that was just one, and that one cost like almost $3 billion. And now I'm getting an email like, hey, you want to come to a meeting? Some of us are getting our genome sequenced. Yes, okay. And so, so it was, the thing was that at this point, so this would have been 2 or 3 years ago now, it was still in a kind of a gray zone. I guess we're still kind of in a gray zone. Maybe we're coming out of it now, but like I could get my genome sequenced, but it had to be only as a medical test. And I was like, okay, so what do I have to do? And so this geneticist said, well, just go to your doctor and give him a sign for this as a medical test, you know? So I'm like, well, okay. And so I go to the doctor, I'm like, could you sign this? And he's like, wait, I'm ordering your genome? And I'm like, yes, it's for journalism. And he's like, I never order a test I'm not totally confident in. And I've never done this before. I'm not doing this. You know, you don't need this. I'm not going to do this. This is not me. I'm not a good doctor if I sign this. So I call up the geneticist and he's like, okay, come, I'll be your doctor. So, you know, he's a clinical geneticist.
Okay.
And he actually like gave me the whole rundown. He's okay, I'm going to just inspect you, like, you know, had me walk, you know, up and down.
So you did a test.
I did a whole clinical workup. Okay. You know, and then they took my blood and sequenced the DNA, and then these clinical geneticists looked at it for these, you know, 1,500 mutations that are known to cause a disease, and I had none of them, and because they're all rare, and that was basically it. They're like, goodbye, and I was like, whoa, like, wait, like, all of that and it's nothing? Like, nothing? Like, this is not right. And so I wanted to get the data because I know that, you know, scientists like you know, you work with genomes. Someone doesn't just come to you and say, oh, there's nothing here, don't worry about it. Like, you have data.
We go digging.
You go digging. So I was like, okay, how do I get this? And they wouldn't give it to me.
They wouldn't give you access to your own data.
Yeah. Yeah. Because it was a medical test. So the raw data, you know, I could not be delivered the raw data as a medical test. It was only the results of the test. So again, I went to this geneticist. I'm like, okay, this is ridiculous. And this guy Robert Green is like, okay, I have an idea. And so he was actually running a study where he was looking at how having your genome sequenced affects your medical care. Does it matter? Because we don't know. Is it worth everyone getting their genome sequenced? We don't know. So he said, I will enroll you in my study, and you will be an experimental subject. And then we'll write a special little, like, extra part of the experiment where you get to have your raw data. Whatever it takes. And so one day, a little hard drive shows up at my front door, and there it is, like the raw data. But of course, you know, it's just a big spreadsheet. I don't know what it means. And so then I had to go.
This is the sequence data.
Yeah, just the raw.
Okay.
Just so
a little bigger than a spreadsheet. Really, really big.
A huge spreadsheet. So your DNA is, if you think of it as being made up of letters, it would be about 3 billion letters. And so if you were to put, people have actually printed it out in books.
Yeah.
You know, it would fill like a shelf going up to the ceiling here. But the thing is that the way that they sequence DNA is just basically to break up your DNA into tiny little pieces, maybe a couple hundred letters long, make lots of copies. It's all experiments. So anyway, that's what I was looking at. Yeah, billions upon billions of these little fragments that just made no sense at all. So then I went to scientists and just said, oh, and they actually enjoyed it because they hadn't, they've been studying genomes of people, but they never looked at the person whose genome they were studying. And so I discovered all sorts of cool things, once the scientists could sort of guide me down in there. And now actually, in the end, one of the scientists said, we had so much fun, it'd be a shame to just throw all the data away. So this is Mark Gerstein at Yale. He said, would you be okay with putting your genome online?
Oh, wow.
Putting our data online. And
so we can Google your data.
Yeah. No, it's all there. So if you just Google Zimmerome, it's there. Plus all the analysis. I mean, I didn't know, should I do this or not? But I just said, okay, fine, I'll just do it. And for a couple years now, Mark has a class where students learn what you do, the bioinformatics.
Yeah.
And he just tells them, okay, here's Carl's genome. Here's Carl. Now I want you to develop a project.
So you're like a digital cadaver. I mean, not to sound more, but in a sense,
nice
students are practicing on you.
Yes, they are dissecting me.
They are dissecting you,
Yeah,
but you're still living.
Yeah, I survive everyone. But they are a little disoriented because they're I'm right there, in the front row. And they're like, Carl Zimmer's Alzheimer's risk, and they slide, and it's fun.
So you talked about
Alzheimer's is fun.
you talked about trying to access your genome, and you in particular were doing sequencing. Which is, extremely difficult as a normal citizen to go and get sequenced, as you have found out. But genotyping, which is another technology where we're not looking at all 3 billion letters, but we kind of have ambassadors across the genome that we can pick. And those you can get from consumer genetic testing,
So
a 23andMe.
So it's about 1 in 1,000 of the letters you're going to get?
It depends. So actually at Illumina, I work on that test and kind of figure out who the ambassadors are for different populations, but it depends on the population and the different products that we offer. But in short, a lot of the consumer genetic companies where you can go 23andMe and Ancestry and National Geographic, we'll use that technology and give you your data for you to download. So you don't have to go through the hoops and bounds. But there's limitations to that too. So I guess the question I had was, have you ever tried one of those companies, and have you done the comparison yet, if you have done it or if you haven't?
I have not done it myself. I just, once I was done with the genome thing, I was like, awesome, okay.
You're exhausted.
Yeah, right. Yeah, I'm going to go through this again, and no thanks. But my family sort of got the bug from me. I'm sort of patient zero, so I can kind of look over their shoulder and sort of see the stuff that they're getting, and it is interesting to see how companies tailor the information for customers, and we can talk about that. I mean, I have very, very mixed feelings about it.
Yeah.
It is interesting then that you can. It is true that from these companies you can get something called a VCF file, and I in the various manipulations of my genome, I ended up with a VCF file too.
Oh, nice. And that's what we can download.
No, you can have the whole thing. I mean,
okay.
You can have the whole genome if you want. It's there.
So the VCF file is not your whole genome. It's just a part of it? Is that what you're saying?
The VCF file that there's that VCF file you can get from 23andMe.
Yes.
This is the genotyping, so it's the 1 in 1,000 basis. And you can, if you want, you can just pump it up into these websites. Like there's one called Promethease.
Yes.
And basically, it just shows you where you fit in with all of the medical literature. It seems, which is, it's interesting, but it's also, you got to really know what you're looking at. Otherwise you might say like, well, I'm going to dye this and I'm going to dye this and I'm going to dye this.
So it's funny that you mentioned that because so the VCF files typically, it's actually quite a new thing. Like when we first started doing genotype, I'm trying to think of, we used to use these two different kind of files. One was called a PED file and the other one was called a map file. And now they condensed it to VCF files, and we use VCF files a lot for sequence data. But Promethease, and there's another company called DNA Land, they allow you to basically take your data and analyze your own data. So you can do a bunch of different things. And Promethease is focused on publications, which has its faults.
Yeah.
But it's also very strong, right?
Yeah, I mean, if someone has published a study saying, like, people with this variant in this gene have this, elevated risk of this disease, that's in their system. And then if you have it, like, it'll just show up on your list, and you can sort of fool around and say, like, oh, I'm only interested in diseases having to do with the heart, for example. It'll just give you that. I know. I mean, there's so much out there, but beware. It might be a study based on like 30 people, and don't even pay attention to it. I mean, I had this weird experience that Robert Greene with Robert Greene was relevant to this. So I asked him after all of this process, and finally he helped me get my genome. I'm like, okay, so I want to sit down with my genome with you, and so he's like looking through in his office, and he's like, oh, look at this one, and it's like something like PRD4, I forget the name, but anyway, so he starts describing what the mutation. I have a mutation in that gene, and he's like, yeah, so this mutation has been associated with this particular heart disease, and I forget the name of it, one of these hypertrophy type diseases. Anyway, basically what it means is like you're walking along as a perfectly healthy adult, and then you jumped it. Like, that's the disease.
That's a great disease.
Well, he's sort of like just talking about it in a very blasé way, and I'm like, why are we not
scared?
He's like, oh, but look at the study, and I'm like, and it's like a real small study, and he's like, we, at Brigham and Women's, they've just been sort of just building up a database of people with all sorts of mutations, and including this one. And after a while, they were like, you know what? Like, having this mutation puts you at no greater risk of having this disease than not. It was just that the numbers were too small before. And even within this one hospital, after they had like 1,000 people with this mutation or something like that over the years, they could say, this is not a problem. But of course, they haven't published that. Like, it's just like they're sort of like, it's their lore that they have. Like, this is what we know within our hospital. Like, looking at our own patients.
Yeah, so it's funny because on Promethease website, there's at least, you go on a website, you sign up for something, and you have a little box, terms and conditions, and you always check it and you never read it. But on Prometheus website, they have like 12 boxes. They're like, this could affect your mental state. They're very thorough, which I find very interesting. I have not done a commercial genetic test. I don't know if it's the same process.
There's a lot of informed consent on those. There are layers of informed consent on those.
Okay, good.
It's not just, are you ready for this, to find out what Alzheimer's variant you have.
Right.
The big one is ApoE4. It's not just the medical stuff, but like, are you ready to look at this genealogical information? Because,
Because you could find out you're maybe not related to the folks you think you're related to.
Yeah, right. You're like, why are all these half-siblings showing up in the system? Like, what? I don't know about these people.
So let's talk a little bit about interpretation, since we're kind of already on the subject of interpretation. And you're analyzing your genome, you're finding all these new things out, and you gave the example of sudden cardiac death and like, what would you recommend? I mean, how did you feel before before you actually knew what things meant? And how did, after knowing what things meant, like learning about the different publications and learning about the nuances, how did it change your view of like reported results? So for example, now if someone says, oh, look, a student comes and, they're looking at your DNA all day, and I'm gonna do it too. and then we find something and, oh, did you know you have the variant for X or for Y? A good example of this actually is pharmacogenomics. So pharmacogenomics is a field where we're looking at variants in genes that affect the way you metabolize a drug. If you could have a response to a drug, maybe you don't respond to the drug at all. Yeah. And it's extremely popular right now because you can do it and it's very common. So like the things you talked about that you found in Mendelian diseases are really rare. In pharmacogenomics, a lot of the variants are pretty common and normal people have them.
So I should mention that in that initial test, a couple of those did come up.
Yeah.
So one was, if I should need to be treated for hepatitis, like there's a particular medication that I shouldn't use because it won't work. And, I'll be curious to hear what you think about pharmacogenomics in general, but I mean, I would say that like it should be like the kind of the low-hanging fruit. Like this should be the stuff that really makes this stuff valuable quickly, because it's like depending on the disease, like you'll see statistics like, oh, such and such a drug doesn't work on 50% of patients. Just because,
Yeah.
And they know that there's something to do with genetics, but they don't know what exactly. But some of these things are starting to come to light, and why it seems so absurd to me that you go to a doctor and they say, let's try this. Let's see if it works.
Welcome to medicine.
Yeah. They're like, doesn't seem to be working. Let's try
this. Yeah.
Like, why waste that time? Like, especially like if you have, say, I don't know, hepatitis. Like, and use a drug that works.
Right.
So I would, but I've heard sort of mixed things about it. Like, it's not yet all that it could be. I don't know what your impression is.
Well, I mean, I would just say in general, any type of result you can get can always be improved and better. And the more we learn, the better it gets. So I always tell people, like, take the result, I don't want to say with a grain of salt, but like, take the result with caution and knowing that it can get better because you contributed, right? With pharmacogenomics, actually, I feel pretty strongly that the things that are currently being used by physicians are quite good. And so one good example is blood thinners, which is like really, really, you kind of need to know which dose you do.
Yeah.
It's pretty critical. So if you get the dose off, you can bleed to death or you can clot up. So you have to get it really, have to get it right. And so we've seen really big advancements in knowing your genotype when you come in and saying, oh, Carl has this variant. We know we have to give him this, and it's very, deadly, right? Or could save your life. But then there are certain ones that are kind of like, so in Asia, for example, there is a lot of cosmetics. And they're looking at, in a way you can consider this pharmacogenomics depending on how you consider a drug, but how people respond to different cosmetology treatments. And, kind of that variation. That kind of stuff I think is very new, not, kind of like the N equals 3 or, not quite developed yet. So you did learn some parts about your pharmacogenomics. Now, do you have it all like written out? And how is your engagement with your physician now, since he said no, and you like found a way, and now you probably know more information about it than he does?
We have not had that conversation.
Okay.
Because I don't have hepatitis, and so I don't want to.
But if you did, he would have gave you the test.
Yeah. Put me on the wrong drug. I don't know. And I do think for doctors, it's that's also a big challenge. It's like, doctors are used to saying like, oh, you have this, you seem to have this problem. We'll test for X. I think you have Lyme disease. Let's test for Lyme disease. And here it's like, oh, let's like give you information on millions of variants that are interconnected with lots and lots of diseases that you may have never heard of. Like, how's a doctor, especially like a family doctor, supposed to deal with that? how do you deal with that with a kid? You know, like, oh, like, do you even tell the parents of the kid that, you know, maybe they have something they're going to have to worry about when they're like 50 or 60? Doctors, I think, our medical schools are going to have to change profoundly, I think, to deal with that.
And I think there have been some advancements. I know at Mount Sinai, and I know some Mount Sinai folks are here, they let the students, first-year students, sequence their genomes, right? The medical students, and let them play around with their genomes. And there's been a lot of stuff that, you know, that was controversial too, even. But so so I guess the question I have is, what was the most interesting thing that you learned about yourself through your genome that you didn't know before?
The most interesting thing, there were a few. I mean, one thing that I guess I was able to use my genome to just sort of understand interesting things about human biology in general. I don't think I have a special genome, but
you do. Everybody has a special genome,
but it's
we're all unique.
So right, exactly. But so we tend to think of mutations as like I was mentioning the letters in a book, and like, oh, if you change one letter, then it can be this devastating mutation, or it can be beneficial. And that is true that genes do mutate that way, but they mutate in other ways, and ways that actually are harder to pick up with these tests from places like 23andMe. So, for example, like you could have a chunk of DNA, and as it's getting as the whole genome is being copied, and that might actually be duplicated. So now you have two sets right next to each other. So you had one copy of a gene, now you have two copies of the same gene. Like, whoa, like all of a sudden, like your cells are just using both copies, and that's like different. And you know, are you making twice as much of that protein? Is that a problem? In other cases, you know, big chunks of DNA can just get chopped out. And so I went to a scientist who specializes in that stuff, and we talked about my genome. And so it turns out that I have a variant in a gene for something called haptoglobin, which is a protein that floats around in your blood. And I have it's sort of, part of it has been duplicated. And what that means is that that's actually, I have higher cholesterol. So on average, you get higher cholesterol if you have the sort of duplicated form than if you have the shorter form. So these things are going to learn about these kinds of duplications and these kinds of mutations. We're going to need, you have to go beyond the 23andMe. And I think that technology is going to catch up so that people will just regularly do whole genome sequencing just as a matter of course. You know, your doctor will just say, like, I don't know what's wrong with you, let's get your genome sequenced. So
did you, in learning that, did you think, okay, now I'm going to cut back on whatever your favorite food you indulge in? How did it change the way you eat, or no? You were like, if I didn't know it before, I'm fine.
Well, so enough, you know, I love my parents, but they've dealt me a few bad cards. I'd say, we all love our parents, but you know, like we have inherited their legacy, and so like there's this gene called FTO, and there's a mutation in there that's associated very much with sort of on average people who have my mutation are a few pounds heavier. And it just has to do with, it's basically a switch for using energy to just burn it off or to store it. I tend to store it. You know, I was in Boston, and this scientist who studies FTO is at MIT, and I'm like, let's meet for breakfast, because I've been looking at my genome, and I have these two bad copies, and he's like, yeah, so do I, and so we go, we have breakfast, and we're. Did
you eat healthy?
No, I didn't realize it. He met me at the hotel restaurant where I was staying, and like at one point I'm like, we both stacked our plates high with the waffles, that, you know, at the hotel restaurants, you know, and with syrup, and I'm like, what are we doing, you know? And yeah, it's so I knew before that I really needed, we all should probably like lay off of a lot of starches and sugars.
Yeah.
And I knew that before. And now I know that like, that's especially true for me, I guess.
Okay.
But,
I mean, to your point, FTO is really, really big, but there's a lot of other stuff, right? And you talk a lot about heredity and kind of like, what do we inherit and what are the things that kind of like impact our genome and the things we don't inherit?
Yeah, I mean, so like height is something really simple. How tall are you? And height is actually very, what scientists say is heritable in the sense that like short parents tend to have short kids and tall parents tend to have tall kids, roughly speaking. And that says to us that genes have a lot to do with it. There's an interaction between the genes and the environment, though, so that even if you have genes that might make you tall, other things being equal, if you have a really lousy diet or if you find diseases all through your childhood, you're not going to be tall. You're going to channel that energy elsewhere. But in terms of the genes themselves, they've associated literally thousands of genes now with height. And genes that don't seem to have, you can't even like make up a story of how they're related to height. It doesn't make any sense, but they seem to be.
That's a lot of us stuff.
Yeah, but maybe they're like connected to networks that are connected to other networks that have something to do with how your cells divide. But just to give one example, like there's this one variant which is like of a gene. It's called HNGA. What I think it's called, and it was like the first gene that was found that's like a common variant that affects height, and it's like the big one. It's the big, powerful impact. And I have one of my copies of a gene that makes you a little taller than average for having it. And I figured it was like putting on a very thin pair of socks. Like that's the on average effect of it. And so it's very hard. You're not going to be able to just go on a Promethease and see that part of your heredity. It's just too hard to sort of make sense of that by looking one gene. Our heredity is too complex than that.
Okay. But kind of talking about, okay, now you have your genome, you're learning these things like FTO, you're learning that you may have certain variants. Let's talk a little bit about kind of like some controversial things you could do with your genome. So I think we talked a little bit about that. And so actually when I think about height, I always talk about dating and height. Because height, to me, like you said, people who have kids, short people have short kids and tall people have tall kids. But I also believe that height, people are attracted to each other because of height. So there's a little bit of selective mating going on there too.
That's true.
Right? So what's the fine line between selective mating when you're making a decision and something like eugenics when you're kind of forcing two people to mate, or not forcing, but you have a desired outcome. Maybe we should explain what eugenics is first.
So eugenics was coined in the 1880s by a British scientist named Francis Galton, and he was actually one of the pioneers of heredity. He looked at people's height and other traits and saw these connections between parents and their kids. So he has a really important place in the history of the science, and he was also, in some ways, a really horrible human being. And he was a big fan of eugenics and a flaming racist. And but this is who he was. And so with eugenics, he was convinced that everything about human nature is inherited. And so, we breed chickens and cattle and crops, let's do it with people. And so eugenics basically means, breeding well, generating, goodness and through breeding. And so he had this idea that you could, this does not make him a Nazi, in the sense that his idea, what he was wanting to encourage was what he considered people to be, who are of good breeding stock, to have kids.
Mm-hmm.
He just encouraged them to have kids. And then over many generations, we would produce what he called a galaxy of genius.
Mm-hmm.
That was his dream.
Yeah, I didn't realize how I was listening to, you were on Science Versus, and they were talking about this, which is a podcast on Gimlet, and I didn't realize how people actually liked eugenics back then. They thought it was like a positive thing until it was not, right? Which is a lot of our history, right?
Yeah.
And it kind of started with intelligence and trying to okay, let's just keep the human race smart.
Yeah, so Francis Galton had a big hang up about intelligence. I think because he was, his family were incredibly rich, but they made their money on things like guns and the slave trade and then banking, but none of them had gone to the university.
Interesting.
And so now Galton gets into Cambridge, and he's like their great hope for their family. This is what it's all been for. And so there's this incredible pressure on him to excel, and he's going to do math, he's going to take the hardest math exam, and he just can't do it. He can't do it. He has a nervous breakdown, and he keeps trying, and he's amazed at the students who can. And he's like, and he notices brothers tend to do really well, or a student and his father both do well. And he starts to get this idea that it's hereditary. And he just, after a while, just drops out. His dad dies. He's incredibly rich. He can just do what he wants. So intelligence was his obsession ever since. And it remains an obsession, especially when it comes over to the United States, where we start to develop intelligence testing. And then there's this idea, eugenics starts to take this darker turn where people say, well, okay, we want to encourage the people we like to have kids, and that happens in the United States. You can see these pictures at local fairs where they give out a prize for the fittest family, along with a prize for the best rabbit and the biggest pumpkin. There'd be like this family, and they'd have a little ribbon. I mean, it's you go to the eugenics tent, and there they are. I'm not kidding. This is just the photographs are just Google away, and you'll be appalled. But the flip side was there was sort of this feeling that the United States was kind of going in the wrong direction, that poverty was rising, crime was rising. How do we explain it? What do we do about it? And it was awfully tempting to say, well, it must be just because these people have bad genes, and so we need to keep them from having children. So that became negative eugenics. And many states in this country passed sterilization laws. Tens of thousands of people actually hundreds of thousands, were sterilized. These laws stayed on the books until the 1960s, 1970s. It was a horrible, legacy of the study of heredity. And the Nazi scientists looked at what was going on in the United States, and they're like, great, we'll take that. That's evidence that we can use to justify what we're doing.
Yeah, so science abuse, abusing science a little bit. Yeah, I always find it's a thin line between what you would say is eugenics has a very negative connotation now, and what you say is selective mating, because I think eugenics is I don't know how you want to define it. It's something that I think we all do as humans naturally. Yeah. In some ways are stronger than others, like slavery. There was a lot of eugenics there. You just talked about the Holocaust was another one. But on the softer side, let's say you're analyzing your genome, and now we have access to a lot of genetic technology like gene editing. And so going back to your FTO variant that has the extra pounds, did that cross your mind at all? The possibility of, using the possibility of kind of newer technologies on the rise and what you can do with your genome in the future, something like gene editing.
It does. It does make it much more direct and concrete, I guess. It's not. It feels less hypothetical. Now, because in vitro fertilization is pretty well worked out. There's a lot of room for improvement, but it's something that there are clinics all over here that are doing.
Yeah, but it wasn't always that way, right?
It wasn't always that way, but since the 1980s, it's been it's been rolling along. So it is very easy now for me to think of in vitro, imagine that, an in vitro fertilization clinic starts sending out brochures saying, hey, we've added on a new feature. CRISPR.
Yeah.
So you donate your eggs and sperm, and we will fiddle with them. Just tick these boxes, and
We'll do the rest.
We'll do the rest. Yeah. And I mean, how different is that from you can go online and find websites for sperm banks, and you can order it up, and again they have the boxes that you can tick off, and we're already. These boxes are kind of weird, I think, because you can tick off religion.
Wow. If I ever do one, I'm lying about all this stuff.
Yeah. What level of education did somebody have? What hobbies do they have? As if that's in the sperm. So we're already okay with sort of people picking out gametes based on whatever standards they have.
Mm-hmm.
So again, what do you
So you don't think this is that different?
Morally, I do.
Yeah.
But logistically, I don't. It's so easy to imagine. And now that we actually have CRISPR babies on Earth,
yeah,
it has happened already. So I can just imagine just a website where it's, they'll say, okay, here's our genetic report. Carl, I'm sorry, but you have 2 copies of FTO that are associated with increased weight and increased risk of obesity. We would suggest for an extra $500, we fix those.
Mm-hmm. Would you do it?
Would I do it?
Where do you cross the line?
Well, that's a good question. Okay, you might think of that as being sort of, you might think of that as a vanity thing. I just want to look good,
It's,
but
obesity, healthier.
Right, exactly. Obesity is a health risk. It puts you at risk of dying sooner.
Mm-hmm.
So, you can totally justify that medically. And there are the line between what you can justify medically and what seems like cosmetic or even enhancement, very blurry.
Mm-hmm.
I wrote a piece in the Times recently about a hormone we make in our brain called clotho. And some of you have a variant that causes you to make more clotho in your brain than the rest of us. And it's generally associated with protection from a lot of features of Alzheimer's, and it's also associated with scoring higher on intelligence tests, just when people are healthy and and are younger. So you could say I don't want my kid to have Alzheimer's. So please, give them this protective change so that they don't have to deal with Alzheimer's when they grow up. Oh, by the way, this is going to make your kid learn faster and have a better memory and so on. It's going to enhance your child. Oh, it's terrible. No, I couldn't possibly do that.
I heard of some folks who did this to get their kids in college.
Yeah, absolutely.
Ready to do your fun.
Right. You've already spent $1 million photoshopping your kid's face under a water polo athlete.
What's another $500?
What's another $500 just to make that little tweak?
Well spent money.
Yeah.
Well, I think the biggest issue with that is that it starts to kind of, continue on the history of capitalism and class and racism and a lot of those things. But I want to stick on this obesity topic with my last kind of discussion point. So another thing, we're talking about the genome, but when we think about our health in general, we can think about our whole biological profile. And that includes the proteome, that includes the metabolome.
People what the proteome is.
Yeah, so when we're saying just think of, so the genome is what's the word I want to use here? Collection of your genes. Thanks. Proteins. The proteome will be a collection of your proteins. The metabolome will be a collection of your metabolites. One of them that I think has a really strong connection with obesity is the microbiome. And a lot of people, well, it's becoming increasingly popular to now do microbiome transplants because they have been shown to alter obesity. So basically, it's a fecal transplant, if you can imagine that. And getting this fecal transplant, they've shown that certain microbiomes are less resistant to obesity and are thinner. And you can get microbiome transplants. So my question is, have you thought about the rest of your ohms?
I've been curious about that, and there are these services where you can get your microbiome at least analyzed and find out what you've got. And I've been curious, although again I sort of have a feeling that it'll just be like, here's a long list of species that you've never heard of, and just kind of like what everybody else has. So I'm not going in with high expectations.
Okay.
But the whole concept is just amazing because like really what it means is that we have the human genome is like 20,000 genes, and then we have these our bodies are about 35 trillion human cells, each of which generally has that one genome. And then another 30 trillion cells inside of you are bacteria. And they are thousands of species, each of which has its own genome, which is really different than yours and different from each other. And so, I forget the numbers. I think it's like if you were to make a catalog of your genes, we just ground you up and just like said, what are all the genes? Like about 99% of them would not be human. They would be these microbial genes that are doing. A lot of them are doing stuff. They're doing stuff that we depend on them for. They're creating a sort of a defensive barrier on our skin, or they are, breaking down food that we can't do ourselves in our guts and so on. And so yeah. I mean,
So you're going to fecal transplant because you have the FTO variant. Next book.
Yeah. Right. That'll be a fly off the shelf. Carl gave me not meat over dinner. But no. But the thing is that there's been a real sort of how do I put this? There's been a real rush on this stuff. And I think DIY fecal transplants are not a good idea.
Yeah.
People are doing
most DIY.
Sorry, I'm in the church of DIY here, but I think we should talk responsibly about these things. And people are doing it, and they're doing it often for good reasons. That like they have these chronic problems, and like conventional medicine isn't helping them. But that doesn't mean we like drive off the cliffs because other things aren't working. So. The thing with microbiomes is that they clearly work incredibly well for certain things. So like really devastating conditions. So like sometimes people get these infections of C. Clostridium difficile in their guts, and it can kill you. I mean, because they're antibiotic resistant and antibiotics don't work. And I think they've done clinical trials now. Like fecal transplants from a healthy person, they will bring you back to good health within a couple days, like about 90% of the time. Like, it's incredible. So, but the problem there is that people think like, oh, well, therefore they'll work for all sorts of things that have something to do with the gut. And it's just not the case yet. I mean, but it's so tantalizing because like we're starting to learn, I wrote about this in another article, we're learning about how the microbiome has all sorts of weird connections to our brains. So people are thinking like, oh, well, if I just take the right probiotic, that will make me smart or protect me from Alzheimer's and all that stuff again. And
here we go again.
Yeah, right.